Okay, thank you, Andy, and thank you, LPI, for hosting and managing all this stuff. This should be pretty good. So good morning, everyone. Thanks for being here. As Andy said, my name is Don Hood. I'm a recent graduate, PhD graduate from Louisiana State University. I just got my degree back in December. Currently a postdoc at Texas A&M, and contact info follows. I'll come back to that at the end. Could you advance, please, Andy? Thank you. So my research, in my mind, falls at the intersection of three different sort of approaches, three different fields, expressed by the Venn diagram at the center there. Geology, classic geology, with, of course, the tools of remote sensing, which are essential to planetary work, but can also be applied terrestrially as well, and the field of data science. I come from a physics background with lots of data interpretation and statistics, so that has really shaped the sort of research that I do. And I've also done a mixture of field work as well as more remote work. So in the upper left there with the darker green outline and dot is one of my projects, exploring tropical serpentinites as potential Martian analogs. Falling between the fields of remote sensing and geology, this was a field trip that we took about 2 years ago now, and the sort of the post-field work is still ongoing, where we're looking at a series of serpentinite bodies in Sri Lanka, trying to understand them geochemically, understand their origin, which is still not fully known, as well as the pathways by which they alter in the tropical environment of Sri Lanka, and also particularly looking at the biology and the sort of things that can live in the serpentinite soils, which are pretty toxic to a lot of plants. That has high potential for the sorts of things we might need to do to make Martian soil more habitable and compatible for growing plants, and the sort of things we'll need to do once we're living there. The next project is one of my published works, looking at the volcanic history on the Martian surface, and this one falls at the intersection of all three of these fields. The graphic there shows a TAS diagram that was generated from a mixture of gamma ray spectrometer data from Mars, as well as thermal emission spectrometer data from Mars, and Martian meteorites and a couple rover samples as well. But this project combined, of course, traditional geology, nothing more traditional than a TAS diagram, the remote sensing components of gamma ray spectroscopy, as well as data science, where these regions were identified using statistics, looking at the entirety of Mars, and we managed to suss out some trends in that area, suggesting or hinting at its volcanic history and what we can learn about it. The project at the bottom there is another field project. It's actually located in Louisiana, looking at the geophysical properties and particularly the subsurface resistivity of a potential terrestrial impact site. It's a theorized impact site, hasn't been proven yet. But the little diagram there shows the resistivity of the subsurface there. And that was a field trip. That particular survey was done in 2019, and the earlier survey was done in 2017. And that's been something I've been involved in. It's more, as expressed on the diagram, more geology and data science, not so much remote sensing, since we're doing a lot of geophysical work. But it's very exciting, and I'm excited to keep moving it forward. The next project on the lower right there in purple is my other published work, again, at the intersection of these three fields, looking at the geochemical correlations in Martian soil at a global scale. This used a lot of gamma ray spectrometer data, hence the remote sensing component, but we used some principal component analysis and other statistics to draw out some less than prominent correlations in the soil, and learn something about where the hydrogen is bonding to soil components and the mineralogy of it. And the last one on the upper right in light blue is something I've been working on for some time, developing an automated system to detect boulders on the Martian surface. And this falls neatly in the intersection of remote sensing and data science. We're just at this point developing the algorithm, finishing really, and figuring out how to accurately and properly detect these boulders. And then as we apply things to it, as we apply it to the surface, we will get into the geology part and actually do some interesting science with it. Could you advance, please, Andy? Thank you. So all of this research that I've done is backed up by the sort of corollary research, the things that you do in addition to research, funding and proposing things like that. I have one funded crowdfunding campaign, which was an interesting experience related to the work in Sri Lanka that I mentioned. 2 publications in grad school, as well as 2 funded assistantships that were proposal-based from the Louisiana EBSCOR funded program. 3 successful field trips that were planned, twice in Louisiana on the left and right of those 3 pictures down there, and once in Sri Lanka, all of which I was responsible for planning sampling strategies, locations, as well as planning routes for the transects for the geophysical work. And I've also served on 4 NASA review panels and been a panelist on one, which has given me great insight to the proposal writing and proposal winning, and what makes good proposals, what makes bad proposals. And with that, Andy, if you could come back to the first slide, that is the briefest summary I can give of my research experience. And thank you all very much for your attention.
Great, thank you, Don. All right, Anastasia. Anastasia's up next. Can you go ahead and start? You don't have to start your video, but it should be able to now.
Okay.
There you go.
Okay.
All right, so whenever you're ready.
Okay, sounds good. Hi, everyone. Thank you, Andy, for the introduction. My name is Anastasia Yanchilina. I will start with giving a brief background of my academic pathway. I will proceed to talk about my current work as a postdoc at the Weizmann Institute of Science, and I will finish with a few words of where I would like my work to grow towards. So I received my bachelor's degree at the Creighton University in atmospheric science and meteorology, where I looked at changes in monsoonal cycles in West Africa, and I related this to decadal changes in global sea surface temperatures over the past 100 years. That led me to become very interested looking back further in the climate record, and I decided to pursue a Ph.D. at Columbia University in isotope geochemistry and oceanography. There I looked at changes in deglaciation of the Black and Marmara Seas over the past 30,000 years, and I have 2 papers from that show different floods that have happened at different times that were really big events. And currently I'm doing my postdoctoral work at the Weizmann Institute of Science in isotope geochemistry, where I largely look at the diagenesis of silica within marine sediments and also fluid rock interactions. Next slide, please. Thank you. So I will start with the diagram on the upper left. So currently in the modern ocean, siliceous microorganisms such as diatoms and radiolaria and organisms such as sponges take up dissolved silica from the water to make their skeleton shells. With time and temperature, these become a sedimentary rock called chert. Now, understanding these phase transformations is important, as the oxygen isotope compositions of chert have been used to indicate the past history of ocean temperature through time, from 3.5 billion years ago to present. And it's been shown that the seawater temperature has largely cooled. Now, the silica O18, it reflects the O18 of the water and the temperature in which it formed. And what I did for my work here at Weizmann, and these are the 2 diagrams on the right-hand side, was measure O18 of all of the silica phases from their initial form of the siliceous microfossils as amorphous opal to the phase of chert through the transition phase of opal CT. And I looked at 2 different types of sedimentary environments. In the first one, this is the figure on the upper right, in this case, these environments had a lot of co-depositional clays. And my work showed that the phase transformations will happen deep within the sedimentary column and at high temperatures. And this is in contrary to what happens in sediments that have a lot of calcium carbonates, and this is the middle figure, in which the phase transformations will happen at much lower temperatures and also shallow within the sediments. Now, going back to the figure on the lower left with the O18 of cherts, the implications of my work are that I'm showing that the silica O18 is a diagenic signal, unless it's the amorphous of what is formed, and it will not really reflect anything original about what was the environment that it was forming, and this is a genetic signal. And I have a paper that came out this year about it, another paper submitted, and one that is currently in prep. And doing this work, I got very interested in understanding different mineral formations, their chemical makeup and isotopic makeup, and really looking into what they say about the original environment of formation, both through Earth history and on early Earth and in extraterrestrial environments. Some of our earliest indications of life on Earth have come from siliceous deposits such as cherts and geyserite. Anyways, next slide, please. So I'm currently looking for a second postdoc in planetary geochemistry in the areas that I described. I really also would like to participate in planning future planetary missions. The list of skills that I've acquired to this point are on the lower left. My contact info is right there. I look forward to talking to anyone who wants to talk to me in the break room or otherwise contact me. And I really want to thank the organizers for putting this together and LPI. And I hope everyone stays happy and healthy. With that, thank you very much. Bye.
Great, thank you, Anastasia. Y'all, maybe you're noticing there's a bit of a delay now when I advance the slide for you. Yeah, it started happening yesterday, so I'm here, I'm listening. All right, up next is Jeff. Oh,
okay.
great.
Yep, can you hear me?
I can, go ahead.
Okay, so my name is Jeff Berger, and I'm currently a postdoc at NASA Johnson Space Center. And my main research interest is geochemistry of the Martian surface. And I work with rover data and do analog studies. So if you go to the next slide, this is an overview of my experiences. I got my Ph.D. in geology at the University of Western Ontario. That was in 2017, and since then I've been a postdoc at the University of Guelph. That's in Ontario. That's where the APXS team is located. And then after that, I moved on to a NASA postdoc program fellowship. I'm at Johnson Space Center, and my advisor is Doug Ming. I'm just about to finish my first year on that, so I have 1 year left. Potentially 2 years left on that. So my research experience, mainly it's about rover and field work. I've been a collaborator on the MSL science team since before the mission launched, helping calibrate the alpha particle X-ray spectrometer, the APXS. So since the mission's been going on, I've helped support rover operations, working as an APXS payload uplink, downlink lead. And we also have a role called the strategic planner, which just makes sure that the APXS is not being used as a shovel, or that we're meeting our science goals. Also done a lot of analog field work. My dissertation has a big project on Kohala and Mauna Kea in Hawaii. I've also worked in Iceland, Utah, Eastern Oregon, Alberta, and Yellowstone. So, because I think it's really important to use Earth analogs to fold into our understanding of the Martian processes. I also have analytical experience. It's pretty typical for geologists: SEM, microprobe, powder XRD, micro XRD, mid-infrared reflectance and emittance, XRF, also PIXI, APXS, ion chromatography. And my recent publications give kind of a good window into my expertise and experience. I published this year a paper on using PIXI, which is particle-induced X-ray emission spectrometry, which is comparable to APXS to analyze Hawaiian volcanics as an analog project for understanding especially the matrix effects that you get with APXS and doing in situ Martian samples, because we can't make powders. We have to analyze things as is on the surface, and there are implications for analyses using that technique. I've also recently published a paper on zinc and germanium enrichments. The APXS found these really remarkable enrichments, 10 to 100 times what you'd expect for a well-behaved basalt. And the interpretation I put forward is that this is a fingerprint of hydrothermal activity. And you can also use the two elements as a tracer for diagenetic mobilization in the sediment. Current projects I'm working on currently, manganese in Gale Crater. There have been recent works that say that it's highly enriched, but it's actually depleted in the bedrock and only enriched in highly localized places like veins and coatings. So I'm working on publishing work on that. I'm also looking at Hawaiian analog material to look at the mobility of phosphorus and potassium, because those are both readily detectable elements on the surface of Mars, and they can tell us a lot about fluid alteration on the Martian surface. So I'll wrap it up with the last slide. This is my contact information. Feel free to contact me about anything. Also, if you have questions, if you're interested in the NPP program, feel free to contact me about that as well. And that's it.
Great. Thank you, Jeff. Okay, next up is Stephanie. Okay. All right, you're still muted.
Here you go.
Right. Okay, whenever you're ready.
Okay. Hi, everyone. Good morning. Thanks for coming out. My name is Stephanie Suarez. I'm a PhD student in geology at the University of Houston. Prior to that, I got my geology and a Master of Geology. I started working on Martian meteorites during my master's degree. Here's my contact information. What I'm hoping to come out of this opportunity that you have gratefully created is, making connections for possible internships or postdocs down the road. I'm a first-year PhD. I'm just getting started, but earlier the better. And I'll be talking about my research during my master's and my PhD. I've been looking at the chronology and petrology of olivine ferric shergottites. Those are a particular type of Martian meteorite. They're basaltic. And I've been looking at a suite of these that have an ejection age of about 1.1 million years old. Ejection age is when did it fly off of the surface of Mars. Next slide, please. Okay, so my master's, I was looking at a meteorite named Tcent. Tcent is pretty well studied because it is one of five witness falls. So of the numerous Martian meteorites we have, not a lot of them were a witness fall that were collected immediately. In that, we have a ton of data on this meteorite. However, when it came to assigning a crystallization age, we had two distinct different answers between three different institutions. And the difference was pretty big. It's about a difference of 120 million years old. So the hypothesis is if Tcent is connected to all these other depleted shergottites by ejection age, who's to say that it's probably from the same crater? Perhaps Tcent, the reason why they had these two different ages is that it represented two different lava flows that were stacked on top of each other that got ejected and travel paired. And so my master's, I was examining 7 different pieces of Tcent. Tcent fell as a strewn field. It wasn't just one cohesive meteorite. So it was lots of different pieces. And I used lutetium, hafnium, rubidium, strontium, and samarium. People were also interested in Tcent because they found enriched components, and these enriched components were ones hypothesized to be Martian soils, perhaps. So we were looking at the lead isotope systematics of that, and instrumentation I was using during this time are MCICPMS, QICPMS at the University of Houston, and TIMS at the University of Wisconsin-Madison. Next slide, please. And here's my PhD project. I am looking at 5 other olivine ferric depleted shergottites within the suite. These meteorites were chosen, one, because they don't have a crystallization age on them yet. We find these meteorites faster than we can analyze them. So that's where I'm coming in to assign a crystallization age to them. Also, I'm adding to my experience and being a hardcore isotope geochemist to now transitioning into petrology. I'll be examining the petrology of these as well. I started on sample NWA 6162. My poster on that will be uploaded soon, and here's just a figure on the left of that. It's cross-polarized image and a element map. And again, I'm like, even though beyond our hypothesis of these being from the same crater, it's just important to add this data to create more groupings within how many Martian meteorites we have. And yeah, I've been branching out now and using different instrumentation at NASA JSC, and that's about it. And here's my contact information again if you want to get into contact with me, and that's about it.
Thank you, Stephanie. And up next is Laura. Should I get you? There we go. Great. Very good. All right, whenever you're ready.
All right, thanks. I'm Laura Fackrell. I'm a Ph.D. candidate at the University of Georgia, working with Paul Schroeder as my advisor. My research focuses on geochemistry and geomicrobiology of Earth and other planets. I'm planning to defend my research in January, and so I'm here today to kind of present some of my research and hopefully make some connections to help me move forward into that next phase of my career. Today, I want to focus on 3 aspects of my dissertation project entitled Applications of Critical Zone Science in the Exploration of Mars. I'm defining Earth's critical zone as a space from the top of the trees down into the bedrock where meteoric water has influence. In this zone, complex chemical, geological, and biological interactions occur, and critical zone science tries to approach these in an interdisciplinary way to understand the system as a whole. Using this approach, I'm also trying to apply some of these tools to the study of geochemistry and the potential geomicrobiology of Mars and how these concepts could apply to agricultural applications. The first aspect I want to address is the development of several Martian regolith simulants. When designing this project and seeking for suitable materials to use as a Mars-like growth medium, I found that Martian simulants that were available in sufficient quantity for growing plants actually lacked several of the characteristics of agricultural applications that I was interested in examining. So the first phase of my dissertation was spent developing Martian simulants that reflected these characteristics. These simulants were compared to data from Mars to ensure that they are actually comparable to Martian regolith. In particular, we did X-ray diffraction, refiltrate refinements, as well as Mossbauer spectroscopy and reflectance spectroscopy. The images on the left show the X-ray diffraction results, as well as some images of the simulants themselves on the bottom left. And this data has been submitted to ICRIS as a publication. The second aspect I want to mention are the plant pilot growth studies I'm using to provide a baseline of plant response to Mars-like soil conditions. The simulants present many conditions simultaneously, and so before you can actually grow a plant in the simulant, you have to provide a baseline of plant response to individual conditions, including salinity. Salts can be rinsed out of the Martian soil before you use them to grow plants, but because water will be a limited resource, it's useful to know what level of exposure to salinity plants can tolerate. The image on the top right shows that nutrient levels in plants after exposure to several sulfate salts. The plants could tolerate much higher levels of magnesium and calcium sulfate, but the sodium sulfate was very limiting to their growth. We're also doing other trials with chloride and perchlorate salts, and all this is part of a publication that's in prep. The third aspect I want to discuss is the use of microbiology in both agricultural applications as well as modeling the potential habitability of Mars. I'm using microbial inoculants to improve plant growth in response to the simulant conditions. The image in the bottom center shows initial studies with these microbial inoculants. I'll also be using Geochemist Workbench to construct theoretical models of how the inoculants affect the mobility of nutrients in these simulants. And these models can be used to inform more complex modeling that looks at the potential habitability of ancient Mars. I'm going to be presenting these results at Goldschmidt this summer, and I'm hoping to submit a white paper on this topic as well. Next slide, please. In addition to these particular aspects of my dissertation, I wanted to briefly mention a few other projects. As an intern at Johns Hopkins Applied Physics Lab, I mapped sulfate deposits on the eastern side of Valles Marineris, and I was able to apply several remote sensing software applications, including ArcMap and ENVI, to produce mineral maps of the study area, some of which can be seen in the image on the left and are part of a publication that's in prep. On my master's thesis, this involved research on the microbiology in hot springs found in Kamchatka, Russia, and microbes, how they influenced their geochemical environments, as well as how they were influenced by their environment, kind of that back and forth. This included techniques such as RNA extraction and polymerase chain reaction for the genetic side of the story, as well as X-ray diffraction and refelt refinements, and a lot of different geochemical measurements in the field and in the lab. And some of those can be seen in the image on the top right. I also have significant teaching experience and mentoring experience through my teaching assistantship with the Writing Intensive program at the University of Georgia. This program teaches students how to write in their specific discipline. I was awarded the Outstanding TA Award this semester, which is given to like the top 10% of TAs throughout the entire university in all programs, and that was a really big honor. Next slide, please. So in the future, I want to continue to use these skills that I've acquired to continue to explore connections among the chemistry and the microbiology and the geology, both in Earth systems, but also the potential for such on other systems and other planets, including Mars. I'm grateful for the opportunity to talk with all of you today. And if anyone has any other questions about any of the projects I just discussed, I hope you'll join me in the breakout session, or you can contact me in the email included in this contact slide. Thank you.
Hey, thank you, Laura. Mm-hmm. Hey, and then Chad, go ahead and get your video going. All right. All right, whenever you're ready.
Hi, everyone. I'm Chad, and so I'll start by saying that I'm a very early career researcher. I'm looking for PhD programs, and so I'm looking for things, like planetary science or geobiology, but really anyone that meets my research interests or is interested in me. So my interests, just to start off, are Mars exploration, life detection, biotechnology, and limits of habitability studies. So I just finished my undergrad this December at Penn State. I got a bachelor's in astronomy and astrophysics, and I had minors in astrobiology and physics. So I have a strong background in math and physics and statistics and coding as well. And I also have some core coursework in geobiology, geoscience, and chemistry and astrobiology. I had some courses taught by Jim Casting and Chris House, and so I had Chris House as my undergraduate mentor. And so with Chris House's lab, I worked on investigating RNA polymerization in an origin of life context, so as an informational polymer, of course. And so we were performing experiments under early Earth conditions with an anoxic chamber and with highly reactive prebiotic components that would have been present on early Earth to try to develop an activated molecule that would polymerize easily into RNA. And so I used phosphorus 31 NMR experiments that I performed and analyzed the data for this work. And so then that led to my summer internship at NASA Goddard this past summer, and I was with the Planetary Environments Laboratory. And so the Planetary Environments Laboratory works with sample analysis at Mars, the GCMS unit aboard Curiosity. And I was working on a derivatization experiment, a pyrolysis GCMS derivatization experiment that the rover is capable of performing. And I was optimizing that experiment on the ground with a flight configured benchtop. And we're trying to look for amino acids, and specifically in Martian phyllosilicates. And so we were using clay analog samples that were doped with glycine, and we were doing experiments in order to maximize the amino acid recovery, changing parameters on the experiments. And so this information we used to advise mission decision making for future SAM experiments that will happen in flight on Mars. Next slide, please. And so now I'm back at Goddard, and I'm working with the Laboratory for Agnostic Biosignatures, looking for biosignatures that aren't necessarily Earth-like. And I'm working in the astrobiology analytical lab. That's the physical lab that I'm working in, the team that I'm working with. And we're performing these extractions and analyses of brine samples from the deep subsurface, 1 billion and 2 billion year old water samples, and we're trying to find amino acids in these samples and analyze them in order to determine whether or not they're due to low biomass microbial communities, or if it's abiotic organic chemistry that's happening at these depths. And so in order to do that, we need to develop untargeting desalting protocols for these really, really high salinity brines, like 12 times ocean salinity. And so it has to be untargeted because with agnostic biosignatures, we're interested in looking at the different ratios of all the biomolecules present. And so we're using a number of different techniques that don't bias the recovery of individual biomolecules. So resin column desalting, as you can see there in the photo, liquid-liquid extraction, and freeze drying. And so we're going to combine those and work on a method that utilizes all of them. And so I've used UPLC fluorescence and time of flight mass spec with AccuTag derivatization for this, and using protocols specifically designed for extraterrestrial samples such as meteorites. So things designed for trace organics, trying to get high recovery with small sample sizes. And I've also performed MALDI laser desorption ionization mass spec in a MoMA-like configuration. And so that was for a polymer complexity study, as well as for sample benchmarking and characterization of field samples. And so some of my additional experience is also doing EGA and GCMS flight data analysis with SAM, and then cryogenic equipment safety and clean room protocol, as well as some GCMS lipids analysis for biological samples. So if you could go to my last slide, please. So this is just some contact information. Just wanted to mention again, I'm seeking PhD programs, Mars exploration, life detection, biotechnology, limits of habitability are my interests. So if you're interested in talking to me, contact me at one of my emails, or talk to me in a breakout room. Thanks, everyone.
Okay, thank you, Chad.